Our next talk is by Martin Borsch Jensen from Gordian Biotechnology, and he's talking about an in vivo screen system to identify therapeutic targets in HFpEF.
Okay, great. Thank you so much. And yeah, thanks for the opportunity to speak here. I'm not a hard person. I'm more of a biotechnologist, so I'll do my best to answer any questions. But hopefully this approach that we're using at Gordian will be interesting to the people in the audience. I'll start out with the acknowledgement so I don't forget. I'm here speaking, but this is really the effort of our whole team, especially the cardiac team. We have multiple indications we're working in. And then we also have wonderful advisors. You heard from one earlier, and they've helped us a lot. So this is sort of an overview of what is it that Gordian is doing. We call it mosaic screening, and basically it's a method to test in vivo simultaneously in one animal a range of different potential therapies. These are AAV gene therapies that we design to up or down regulate some gene or a non-coding target, and that contain a barcode so that when we have a pool of these different AAV gene therapies, each of them is sort of uniquely identifiable. We then inject those into an animal together at a low dose. And so we're not trying to saturate the tissue and get every cell perturbed with something. We're trying to get sort of a single-digit percentage of the cells as having some perturbation. And those are, you can think of them as sort of, okay, instead of doing a 96-well plate and we have different independent experiments separated by plastic, these are independent experiments that are separated by diseased tissue. And so that means we get all the complexity of disease biology, whether that's interactions with the immune system or metabolism, et cetera, and we can see what these perturbations do in that context. The independent part is tricky. I'm sure I'll get a question on that. We can address it later. Then we use single nucleus RNA sequencing as a phenotypic readout for what did each of these perturbations do. And so we can take out the tissue, pull out the cells, sequence them, and then assign each cell or each nucleus to one of the interventions that were in the pooled library using the barcode. Then we can pool many nuclei that receive the same intervention, and then compare those to various controls and to our priors across many different, here I just put features. So these are gene sets similar to what Virginia was speaking about earlier. And so that lets us pick ones that have potentially therapeutic effects, and we then follow up with sort of a normal experiment where we just take that single intervention and inject it into animals. And of note here, you can see a mouse, rat, something, but we can do this in any animal, more or less. We do work with mice and rats, but we've also screened in horses and monkeys, and it depends on the indication and what the best model seems to be that we can access for that indication. All right, so for the stuff I'm about to show, these screens are done in ZSF1 rats. You're pretty familiar, so I won't spend much time here. We phenotype the animals. We inject the AAV at 4 months of age, and we phenotype them just to know that we actually have a disease. And then we leave the AAV in the animal for 4 weeks. The hope here is that we see not the first order effect of perturbing something, but rather what would happen in a patient once the whole tissue has responded or the cell has responded to whatever we did. Here's one library that I'll show you. This is 151 different targets. You can see it's across a bunch of different biology. I like to say that we sort of invented this mosaic screening so that I don't have to be smart and know how HFpEF works. I can just test all of the things. We do both up and down regulation of genes, and we can do secreted factors or non-secreted factors. All right, this slide is a lot. We'll spend some time here. Mildly butchered by the intensity of the light, but it's okay. So what you're seeing here, each column is on the order of 1,000 nuclei that have one of the interventions. So every single column is a different perturbation gene up or down. And then every row is a gene feature. And so these come from a variety of sources. Some of them are sort of expert guided, like, okay, we know that hypertrophy is important, these genes are relevant. We also derive features in a sort of unbiased way from looking at patient samples and our animal models and a variety of sources. And so over on the right, you can see, if I listed all of the names of the features, you couldn't read them anyway, so I've just clustered them into broad biological categories. And you can see we've indicated here's our desired direction, so that means, okay, these genes are related to sort of ECM deposition, we'll want those to go down, and so on and so forth. So you get a whole lot of information, as you can see. And we see some clustering where some genes have similar effects, but there's a lot of variety going on. And then in the bottom, you can see we have, in the middle of the map, with the blue arrows, we have our inert controls. So these are interventions we put in that are supposed to do nothing. And then there's a few positive controls. So these are things that are in clinical trials, they're in red. And then just for indicating, we have a bunch of things that we chose to follow up on in black. I won't get to talk about all of those, but yeah, some of them you can see have been selected because they have really strong impacts on calcium metabolism or calcium handling, and others will be sort of like lipid metabolism and so on and so forth. So this is the output of one of these screens. It takes quite a bit of work to generate. Then we follow up, as I mentioned, and here we use both the ZSF1 rat model and the 2-hit mouse model. For the mice, they're at 20 weeks usually, and the rats are again at 16 weeks. And so we do a whole bunch of characterization, all the stuff you'd expect. I won't dwell on this. To give you one example, here's one set of data from one of these follow-up. It says target 1, but I can tell you that this is CBIN1, which has been studied in Utah. So this is not like a Gordian original, which is why I'm telling you. But you can see that, okay, we have an improvement in the diastolic function. It seems like we also have improvements in hypertrophy, potentially. And we have our sort of LV mass by echo, and it correlates very well with the overall heart mass. We have no change in ejection fraction. So this is sort of an example of the kind of standard data you're familiar with from one of our assays. And so what I'll show you additionally is that because we have all this data, we get to nominate a bunch of targets. We can actually combine these validation studies with all the transcriptomics we have in order to apply machine learning and make new predictors for the physiological phenotypes that we actually want. Because at least for me, I don't know how HFpEF works. And so that means we can try to sort of pick the targets based on this is the desired directionality, but we're not 100% confident in that, right? So when we combine these different data sets, we can take, this is just an example with E over E prime, but we do this for a bunch of different physiological things. We can take each dot here represents a whole study with one target compared to a control injection, which is just AAV not expressing anything. And then what's the actual effect we see? And then we can do, again, I won't get into all the computational stuff. There's like a lot there, but let's just boil it down to machine learning. In order to create a predictor that looks at the transcriptome, and then says, okay, I think that E over E prime will move in this way for this particular target. We can then compare that to what are we actually getting. Here it's just at a 5-week time point, but that's arbitrary. And then we can make these predictions, and we see that we can actually do this fairly well. Like we have pretty high predictive power, which means we can do these screens for 150 targets and then pick 10 or whatever, where we think we'll have certain physiological outcomes. This is rat screen and then a follow-up study in rats. But if we then take the machine learning train just on the rats and don't retrain on mice, and then see how well it predicts the physiological outcomes in the mouse model, it's still pretty good. So that's good. It seems like we're picking up things that aren't exclusively we're overtraining to the model that we have. So we do this, and then we nominate a bunch of hits. We try to validate them. I won't have time to go into all of those, and a lot of the studies are still ongoing. But we do want to look at the positive controls just as a sanity check on how is our stuff working. So we have 3 NPPA or ANP. Lily's looking at that. Bunch of trials in Japan previously. This one is closely related to the I1C that is in trials from AskBio. And this is from Tenaya, obviously. There's more nuance in the colors here. Sorry, it's like a little bit blown up. But basically, if we look at the biology that we expect to see for each of these targets, that sort of makes sense. Of course, there's a lot of stuff where we don't know what we're supposed to see. And so we see something. Is that true or not? We will leave the mechanistic studies to others. But it seems to make some sense. And if we then go into, okay, our more machine learning approaches, what is it that we're predicting? We're predicting from those that overexpressing NPPB or NPPA or this one should reduce E over E prime largely by increasing E. And that's sort of consistent with, and I probably don't need to spend time on this for this audience, but that's sort of consistent with things that have been observed in clinical trials, even though it's somewhat limited, right? They only did a phase 1 here. They do see these changes, but at low N. So in conclusion, we have this system that we think will allow us to do high throughput in vivo validation of so many different hypotheses. And hopefully that can help us find therapies that work well, even before we at least understand the disease super well. So what's happening now, we're running a whole bunch more screens. And then we have ongoing validation studies for things that were identified in the first screen that I showed you already. So we have some 5-week data. Some of the things look really promising in terms of this one even reduces body weight, so bonus points, but improving diastolic dysfunction. And we're waiting on sort of the 4-month data for those. And that is it. Open for questions.
Nice approach. Really great to see a really unbiased way of addressing this really important question. Simple question: your AAVs, you do not include an shRNA, and I ask because it'd be nice to get that directionality to further validate the hits, not just go in with increasing a gene, but also go in and reduce genes and et cetera. So I'm just curious.
Yeah, we're doing both. So the library here was like 80% up, 20% down, but we have both sort of RNAi and CRISPR that will take things down, okay. And then we also have overexpression. Yeah.
Great work. Can you imagine how much is cost of this kind of screen?
So, a few $100,000 per screen.
So I'm curious. So you have a readout single nuclear RNA sequencing. So to some extent, you can pinpoint the barcoding to a specific cell type, right? And if I understand correctly, in the heat map, you kind of put together the features in agnostic kind of cell. I'm wondering if you can prioritize targets based on the cell type where I reach, more in this direction. You are ultimately, if you are taking more a cardiomyocyte focus approach, or you're focusing more on different cell types, you find these regulated targets.
Yeah, I should have mentioned this. So we tend to focus on 1 or maybe 2 cell types at a time, because otherwise you're trying to do all by all. And then when you have different numbers of each cell type, then you end up with like, okay, we need to do 20-fold more so we can get enough pericytes and so forth. So this all was cardiomyocyte specific. You can use a promoter in the AAV to make it cardiomyocyte specific. And the disease biology we looked for consequently is cardiomyocyte specific, except we are interested in these part of being in vivo, or the point of that, right, is that we can't tell this is a macrophage gene that's up if it's not looking at a macrophage gene, but we can look at the second order effects, right, of what is a cardiomyocyte that's in a more inflamed environment or a more fibrotic environment and so forth. And so we're creating, with these sort of indirect approaches, we're creating predictors for what is the environment of this particular cardiomyocyte.
Thank you.
You mentioned this briefly in your talk, but can you speak a little bit more about what you mean by independence, especially for things when you're modifying secreted proteins or plasma membrane, transmembrane receptors or channels or things like that?
Yeah. So it all just gets down to sort of brass tacks, do the experiments to test for independence. So there's no magical thing that creates independence. It's just bring your dose low enough. And so we've done a bunch of studies to calibrate on what is an acceptable dose. So for example, we take a sort of known desecreted factor that does stuff. We inject animals with different doses, and then we see at what percentage of cells transduced do we actually start to see in the non-transduced cell signatures of this particular protein. And then we go 100-fold below that. And so I wouldn't guarantee 100% independence, especially sometimes 2 cells will be transduced next to each other, but then you're sampling over 1,000 cells, and so you sort of average that out.
Thanks for this talk. I have a question about kind of target subtypes. So in your example, you have NPPA and then also looking at HDAC6, and this approach seems really great to find cardiomyocyte and autonomous targets. But when you're talking about, say, natriuretic peptides that may influence whole body, like natriuresis or blood pressure, how are you thinking about kind of the flavor of targets and how it's influencing the transcriptomics of a single cell?
Yeah, there's a lot of stuff there because also obviously you're thinking about what's the drug modality here and like how will we deliver this and so forth. One advantage we have is that I showed HFpEF here, but we're also working in sort of liver fibrosis, lung fibrosis, and osteoarthritis. And so we do get to test the same targets in a bunch of different organs. But I think, yeah, you do your literature search and you might get a hit that like looks good in the heart. And if you then suspect or see in validation that there's toxicity, then you got to find a different way to hit it. The one thing there is if you're using AAV as a modality and you can limit expression to the heart, you could potentially use sort of like a much lower dose such that the local concentration is much higher than the secreted concentration. And so you get positive effects. But yeah, it's something to watch out for. We don't immediately see all of that in the screen.
Okay, thanks.